Hi, my name is Arianna Mathewson, and today I'll be presenting on our research project titled Mitigation of Glyphosate-Induced Developmental Defects in Medaka fish. This project was done by me and my research partner, Drake Weidman. So to start off, glyphosate-based herbicides are the most universally used herbicides worldwide. Glyphosate, an active ingredient in Roundup, has been known to induce oxidative stress in animal cells, including fish. Antioxidants such as ascorbic acid, also known as vitamin C, can reduce oxidative stress by donating an electron to stabilize the reactive oxygen species, or ROS. Our previous studies have demonstrated that environmentally relevant conditions of glyphosate can affect developing Medaka fish embryos, but less was known about potential methods to reduce the negative effects of glyphosate exposure on embryo development. The experimental question we posited was, does the addition of vitamin C to a treatment of glyphosate reduce its toxic effects in the Medaka fish model? So to do this project, first we collected eggs, and eggs were collected from specified fish tanks, and then eggs were processed, and fertilized eggs were chosen for experimental use. Then came the exposure, and for this, eggs were exposed at the same time to ensure consistency throughout the experiment, and exposure treatments remain consistent throughout the duration of the study. After exposure, the eggs were monitored each day under a microscope until 15 days post-fertilization, and observations were recorded in our lab notebooks. Embryos were examined 6 to 8 days post-fertilization, also known as DPF stages, as well as 15 DPF stage to determine if their development was affected by the various treatment groups. For data analysis, after the experiment had concluded, data was compiled and analyzed for statistical significance. And the main focuses were mortality rate, heartbeat rate, hatching success rate, and phenotypic changes. For the phenotypic analysis, pictures were taken under a microscope for the duration of the experiment, and then were analyzed for phenotypic changes from 6 DPF to 15 DPF. We did some DNA-RNA extraction, and for this, DNA-RNA were isolated from the preserved fry samples using the QIAGEN All-Prep DNA-RNA kits. And then nanodrop was performed using 10 microliters of the isolated DNA and RNA samples. And these samples were placed onto the nanodrop and then analyzed for both sample concentration and sample purity. As you can see, for our 6-well plate setup, we had our control, vitamin C, glyphosate, and glyphosate plus vitamin C treatments, and these were run in triplicate. And this was to validate the collected empirical data and observe results for our experiment. For our treatment groups, we have here kind of laid out the ratios of each material for the treatment groups. We have our ARM solution in 100%, then our ascorbic acid, glyphosate, and ascorbic acid plus vitamin C treatment group. For data analysis, we have the mortality rate, and you can see here that glyphosate had a much higher mortality rate than all of the treatment groups, and then when the glyphosate was co-treated with vitamin C, the mortality rate lowered again, and this was considered statistically significant. Next, for hatching success rate, we have our control vitamin C, glyphosate, and glyphosate plus vitamin C treatment groups. And you can see that the hatching success rate was statistically significant for this data set as well. And here you can see that hatching success rate was significantly lower for the glyphosate treatment group. And then these effects were reduced when co-treated with vitamin C. For phenotypic analysis, we have 4 different groups. Criteria that we looked at, we have the swim bladder inflation, enlarged yolk sac, graying of yolk sac, and spinal curvature. So for these, the swim bladder inflation, graying of yolk sac, and spinal curvature were all considered statistically insignificant, but of these 3 statistically insignificant analyses, the spinal curvature was the closest to being statistically significant, which is why we denoted it with an NS and left the other 2 blank. However, the enlarged yolk sac was considered statistically significant in our data set, and we believe that a lot of this was considered statistically insignificant due to the smaller sample size. and would probably have a different outcome if we had a larger treatment group to analyze for statistical significance. In our results, like I mentioned earlier, each treatment was run in triplicate to validate the collected empirical data and observed results from our experiment. Hatching success rate was reduced by glyphosate exposure, and vitamin C co-treatment mitigated these defects. A higher mortality rate was found in the glyphosate group, which was mitigated by a vitamin C co-treatment. The vitamin C co-treatment tended to decrease yolk sac enlargement and spinal curvature defects in embryos, and this was with a p-value less than 0.06 to roughly 0.09. And the study demonstrates that vitamin C and antioxidant can have an ameliorative effect on the developing fish embryos exposed to glyphosate at environmentally relevant conditions. And future steps, oxidative stress genes should be sequenced using qPCR to see if there is a genomic change between the glyphosate and glyphosate plus vitamin C treatment groups. There was some negative effect on the embryos due to the acidity of the ascorbic acid, vitamin C, so it would be beneficial to replicate the study with a lower concentration of vitamin C to see if the results still hold up. We would like to thank the UNCG Biology Department, our principal investigator and mentor, Dr. Ramji Bhandari, Chelsea Smith, whose previous research served as a baseline for the study, and fellow lab members who assisted us in the completion of this project. And I have our reference at the bottom right corner, and this is just a citation of Chelsea Smith's paper, which also studied the epigenetic and developmental defects from exposure to glyphosate and Roundup on developing Medaka fish. Thank you for your time.